Um, I'll be giving a talk on microscopic method uh, in the application of biology, and this is a very vast topic. It can go from a small single molecule level to tissue and beyond. And in one hour talk, it is very hard to cover all type of microscopy technique, but I will be trying to cover as much as possible, particularly the kind of research you people are doing in the lab. So first thing is, what is microscope? So it is a small instrument where you can see very small object what you can't normally see in naked eye. So now it comes into the challenges. So first is what we want is to do enhancing contrast. Then next step is we need and we are developing different kind of innovative techniques. We are exploring different wavelengths in the electromagnetic spectrum. Optics is one thing, but there are other things what I will be coming later. Next is innovative optics and spectroscopic method. It's just not only visualizing things. We can quantitate things, we can get numbers out of it, we can understand the mechanism, how two molecules interact, how cell moves, or any biochemical processes, how it's happening. You can real time see it, visualize it, and that's why it is very powerful. And next is the quantitative biology. You can get a physical number out of it, and you can do physical modeling, and you can understand the mechanism. And that's where the microscopy is going, from the point where it started as in the picture, just seeing an object. From there we're moving towards quantitative biology. So in this part, because I know in IISER students are from different discipline, and it is very microscopy is a field where expertise from different disciplines are required. Like of course you need optics, you need chemistry, because how you tag the molecule, what is the best molecule to address a certain problem. And then of course there is engineering. Any instrument operates well once you do engineering aspects well. Then there is the biophysics, understanding the mechanism, how molecules interact, move, diffuse, any kind of processes, how disease happens, or how development happens into a other end. And then is the computation. And in the recent past, computation is taking a extremely powerful role to evolve the microscopy in different level. And even there are fields like evolving, like computational imaging and all these. So I won't be talking about this much, but I'll let everyone know that even deep learning and any other techniques are becoming immensely useful in biological imaging, and combining imaging with analysis and understanding of the biological processes. So as it is a broad field of biology, so it can explore from single DNA 2 nanometer to protein, which is like a tens of nanometer maybe, and then it can go to cellular different organelles, to go to cells, which is of the order of micron, tens of micron, and then it can go for tissue. It can go millimeter to centimeter, even larger. Like suppose if you are or dealing with human brain, like a whole brain, you have to understand something happening in the whole brain. So you have to deal something in tissues on a very large scale. So everything comes under microscopy. So if you see the electromagnetic spectrum, so it is important to see what are the objects. Like this is visual range. It is cells. And then this is the range where maybe cluster of cells or organoids, this happen, and then is a big like a butterfly and large scale objects. And on the other side, you can go for smaller objects to molecules to atom and beyond. So because the size is mapped, as you can understand, majority of the thing we see when the electromagnetic wave passes through it and interact with it. And if it is wavelength is much larger, you can't see it. So in this case, the majority of the things we can address by visible wavelength. But I just want to mention that there are microscopes in infrared spectrum and using ultraviolet or X-ray origin also. And of course, there is electron microscopy. It's a big field of electron microscopy that I won't be talking here, but everyone should know it. So what is the advantage of microscopic techniques than many other chemical techniques? Of course, every technique has their own strength. Biochemistry has pushed biology very far, understanding of the problem. Then that's why one can also go pushing the microscopy based or physical based techniques. So, but one of the major advantage of microscopic techniques, this is non-destructive, particularly the optical techniques. Not all, but a good fraction of them. So basically, you are not killing that cell or destroying the thing and taking the proteins and everything out of it. You can visualize it, the whole process, even the molecular interaction, everything in real time and without destroying the thing. You can keep the cell or organ and you can bring it back after some time and again see it. Of course, everywhere there is technical challenges. I'm not addressing this, but course-wise, this is the main thing. So non-destructive, and you can see live processes in many of the methods. And any technique can be scalable. Like from the same tissue, you can see single molecular level to cellular level or tissue level, or even if it starts coming from a big organ like a tumor, some portion of the tumor in brain or somewhere, you can take those portion and you can understand whole thing in a very different levels. So and of course, same microscope can't address all the thing, but once you are having the understanding the big problem, you can understand in different level where the problems happens. And then is the quantitative techniques, which is beyond normal visualization. So like we can get many numbers out of it, what time scale it is happening, what length scale it is happening, those kind of things. And everywhere in any one of the method you do, particularly analysis based method, you have to be very careful about the artifact, because you can get some number which may or may not have any meaning. So I won't be talking about level free techniques much, but in a very coarse way, level free techniques are the techniques. So here, so main challenge is what Gandhiji was seeing in a microscope. So main challenges is once we try to go beyond this thing, we don't get enough contrast, because the refractive index of any organelles are very close, and it's almost like a water. So you don't get enough contrast to see these things. So majority of the level free techniques are using the contrast enhancement method. From bright field, it is going to phase contrast, DIC, or dark field microscopy. And there are techniques like a quantitative phase microscope. Like this is another way of huge broad range of level free techniques are coming up. So that is also, I won't be talking in details in this course, but you should also know, like this image in the left side, it is a phase contrast image where you enhance the contrast and you can see the edges of the cell and you can understand where the things are located. But this brightness is don't have any meaning. It is just giving you contrast. But on the other side, you can quantitate the phase information, because once light is passing through different layer of the tissue, there is a change in because of the different index of the material. There is a change in light path, and that if you can capture that aspect, you can get this quantitative phase imaging. So here, if something is very bright, it is higher in depth, and something is dark, which is much lower in depth. So basically, you can get a almost topological map of this thing. And there are techniques like holographic microscopy and other thing I won't be describing, but you get a course idea that level free techniques are also not useless people are doing research on this thing to get a lot of quantitative information. So it is also beyond visualization. So now I want to talk, in this part, majority of the research are what people are doing in the laboratory are fluorescent based techniques. So I'll be mainly giving talk on fluorescent based methods and different domain. So like this MDO development in the left. So here you can see the whole MDO development. It is zebrafish MDO. So on the top, this cells goes and it forms the spine. Here it forms the eyes, like that. So, but this kind of phase contrast based thing, it gives a very cool image, but you don't get information about what level of the cells, what is happening, because it is a combined information of all the plane. Okay, so you don't understand, and if there is a defect in any portion of the tissue, you don't have the information. So from there, you can go to a technique like this image is same thing in transition in fluorescence. This is a zebrafish embryo development. What it is a home-built microscope system we have developed and imaged it. So you can see single cell level how the spine is formed. So completely from there, the transition is happening at different level, because this is a and the same thing you can see in dorsal and also in the ventral thing. It is like a millimeter, whole length scale is about a millimeter. So such a big embryo, you can still track all the cells in single cell level. And if some portion there is defects or something, you can point it out. And if you do any mutation, how the whole method of the development changes, that you can track it down. So you can clearly understand from a normal level free technique to shifting into fluorescence, it is giving away a lot of advantages. So what is fluorescence? Again, I will just briefly talk about. If you there are some molecules which is having the fluorescent property. If you excite them with a proper wavelength, and it goes to higher excited state, and after some time it come down to the ground state. And during this thing, it emits the fluorescent wavelength, and that you capture. So these molecules, if you tag with your molecules of interest, so wherever the molecule B, some fluorescent molecule is molecule B is there, which is tagged with your molecule of interest, which is molecule A. So wherever molecule B is giving the fluorescence, there is presence of molecule A. So you can track this dynamics, you can understand the processes by using fluorescent-based technique. So as I told you, it is a leveling is in different biological proteins and organelles, and it can detect the location in very precise way. You can resolve specific individual structures within cells or within tissue, and you can go for biological processes. And in fluorescence, one can go for fluorophore or quantum dot or expression of GFP, which also got a Nobel Prize a few years back. One major reason is you can not only have genetically tagged cells or tissues, you can also go for animal. You can have fluorescent fish or mice and like that. So you can take the animal and you can image him, and with a very high precision. And there are different kind of fluorophores. I'm just going through it. You are having the presentation, so you can read these details later. So basically, you can have biological fluorophore, quantum dots, which is having a toxicity, one should keep it in mind, and also then organic molecules. Now, which microscope is best for your application? That is the main question what they asked me to discuss. So before discussing it, I want to give this thing. This was in Nature several years back. The good, the bad, the ugly. The imaging fluorescent molecules in live cells is revolutionized cell biology, but pretty images is not necessarily a good one. Many biologists are learning this in a hard way. Basically, any image is giving a very good contrast, very good looking, I mean, it may not be always very good image, because it goes to the next step. What you want to get information, like in this case, it is oversaturated. It may get a very good contrasty image, but this is not a good image because it is oversaturated. So many of the pixel you are losing the information. So before doing the imaging, you should also understand that understanding how imaging is done, understanding that part is very essential, because otherwise you will pick up artifact and give you wrong result and wrong interpretation. So as I explained, there's a different length scale in biology, and here this is a very critical thing. This is the kind of wavelength of light and diffraction limit, because once you focus a beam, you can't make it very small. You can't image a DNA by focusing a laser beam, because it is you can, because laser wavelength is of the order of 500, 600 nanometer in this range, where DNA is only 2 nanometer. So it won't interact with it; it will pass by. So now the challenges is come. So once any object is this region, like 100 to 110 to 100 micron around that region, you can generally see it by normal, very old classical microscope. But once you try to go push the limit to 3D or in millimeter or centimeter range, there is challenges. It's completely different challenge. And on the other hand, the moment you want to beat the diffraction limit and want to see beyond smaller structures than this diffraction limit, you find different challenges. So same technique, a good microscope in this regime can't address your problem in the right regime. So understanding what microscope is, what is your problem and what is the best microscope for it is important to understand. And as I told, left part is the tissue imaging aspect, and right part is super resolution or defined spectroscopic technique, TARF, then FLATE, FCS, and others. TARF is also a super resolution when different. So next is, so first I will be talking about the challenges in the tissue imaging or large scale imaging. Here challenges is the resolution. Of course, it's always the challenge. Like you still want to get a decent good resolution, XY in Z. Particularly, majority of these problems are in 3D. Like if you want to see in 3D how different levels of the tissue are interacting. So here the problems comes in scattering, the autofluorescent, light penetration through kind of any media. Because light, if you put a light on your arm, light won't penetrate throughout, because it scatters so much. After some time, it will decay down. So you can't see very thick object. And second thing is autofluorescence. Any biological molecule, even if you do proper fluorescent marking, once it is in a thick biological tissue, it gives a lot of background, gives you very high because of the autofluorescence, and which makes it more challenging for you to visualize it, because of autofluorescence dominates your fluorescent signal. So, and this is the thing, like a diffraction limit. If you kind of focus a beam, you beat at a tiny point here. This is diffraction limited point, and which defines your lateral and axial resolution. So basic idea is, if you are having a molecule which is moving, which is much smaller than diffraction limit, and you are scanning with a beam which is much bigger and bulkier, which is this one may be of the order of 10 nanometer, whether your beam is like half a micron by micron in Z direction, then you can't see it that small, you will see it that big. That is the main thing. The fluorescence is showing it in a big structure. So Now, if two molecules are closer, like if these two molecules are very close, you will see like this. So you won't be able to differentiate the signal of this green and that green. You will see a combined blob. You won't be able to say, differentiate it. But the moment the molecules get little far, you can start to see the two molecules together because there is a dip in the middle. So that is the main idea of resolution, and same thing applies in Z direction as well. So if you are having an array of protein like this of 10 nanometer, and you are brushing and you are doing a painting with a 1 micron brush, of course you will see that thick one. You won't be seeing the small molecules. You will see this thick brush, this thick painting. But now, if you are using a technique where you can go to smaller one, it is much thinner, and there it comes the diffraction limit, and can we go beyond below it, and can we go above it? That is the main challenge in the resolution. Okay. So to do imaging, particularly in the tissue scale, also means you have to understand another important parameter, that is time scale. So where there is fluorescent lifetime, protein folding, diffusion, I'm not elaborating it, but you can see everything happens at different time scale. So your technique should be able to address the problem where we don't lose the fluorescence, and you can still see your processes. So now I'm covering a microscopy technique from single cell to tissue. There are different techniques. Sorry. Like epifluorescence, confocal, spinning disk, multiphoton, light sheet microscope, all these. So in epifluorescence, what you do is you are sending a beam, and there is a particular kind of mirror which is called dichroic mirror, which reflects the beam, which you are sorry, having a bit cold. So basically, your dichroic mirror goes to your sample, and you are illuminating it. So here, these are the advantages. Sorry. So there is no depth selectivity. It has good temporal resolution. It has low photo bleaching, and it is good for imaging a monolayer of cells, but with poor resolution. You won't be able to see different layers because you are illuminating everything and collecting the emission of all the plane. So there it comes the confocal, the advantages, because in confocal technique, where if you are having a thicker tissue and or even in cells, you want to see layer by layer some cell nucleus, you want to see in each of the layer, you need a resolution in Z, because so what you do is you are sending a so suppose if there is an object in your focal plane, green, and you put a pinhole in the conjugate plane here, such that the beam coming from this one only passes through and collected in the detector, and the beam and the light which is coming above and below are blocked by the pinhole. That is the main principle. So here, of course, because of the pinhole, you are focusing at a point. You are not seeing, compared to other technique, you are not seeing all the planes because illuminating everything and collecting everything. Here you are focusing at a point. So now you have to scan the point to reconstruct your plane and go in different plane to go in 3D. So it tells you clearly, although it gives you XY resolution, but it gives you poor Z resolution. Compatibility poor Z resolution. And second thing is, it gives a very poor temporal resolution, because you are scanning plane by plane and point by point. And so because it is in this technique you are sending the laser, excitation wise it is exciting all the planes upper and below. So the molecules which are fluorescent are getting emitting fluorescence from all the plane, but you are blocking it by using pinhole. So but still it is doing the bleaching, photo bleaching in the upper and lower planes. Okay, I didn't mention the photo bleaching, but maybe all of you know, but still one line. If, as I told in the Jablonski diagram, if molecule is excited, after some time it comes down. This cycle can't have infinite time. Every molecule has some period. After that, it gets oxidized and loses the fluorescent property. So that once it loses the fluorescent property,
sorry,
photo bleaching.
Dipanjan, can you remove that or hide that meet.google.com is sharing your screen? We are unable to see part. No, right. Thanks.
Sorry. I didn't realize it is also coming that side. Okay. So so this one is good for tissue imaging, but of the order of a few hundred micron. Beyond even 100 micron is tough unless the tissue is very transparent. Like around 50 micron is a better one, I would say. But beyond this thing, normal confocal is also not good. So so this is the main geometry. You send the beam, and there's diaphragm mirror. The laser passes through all the planes, but you are collecting the fluorescent from a tiny point, which is this point, and through the pinhole and collecting on the detector, and you are scanning the beam throughout the sample X, Y, and Z, and you can reconstruct the image. So actual microscope is more complicated, but that's why I do this very simplistic diagram to understand the pinhole concept. So such that you are just taking from one plane, you are detecting planes upper and below the fluorescence, but till you are exciting all the planes, so you are doing photo bleaching all the time. So photo bleaching is high. You are having poor temporal resolution, but you gain in a good XY and moderate resolution. So now it comes this comparison. You can see it. I'm just skipping through what I explained already. And then there is a technique called spinning disk, which is very similar to confocal. So rather than one pinhole, you are having an array of pinhole and you are scanning it. So basically, you are enhancing the speed, but between the pinholes there is a crosstalk, so you lose a poorer XY resolution compared to confocal microscopy, and also in Z, resolution is not as high as much you can achieve in confocal microscope, but still it is a good compromise because you gain in temporal resolution very much, particularly important for the thick and big tissue. Then it comes from another thing, is multiphoton. Although it is, I wrote it like a fluorescent technique, like a visual wavelength, but the excitation part is not visual here. In a normal confocal, you are exciting, as I shown in the earlier diagram, the beam, you are sending a beam corresponding to this wavelength, and you are exciting the molecule. But rather than that, you are giving half the energy in a very short interval in the same position, so that the molecules, same molecule can get the similar amount of energy. So this goes in infrared, near infrared regime, and infrared to near infrared in that regime, and so that it can go to excited state, and after some time it kind of emit, and you get the fluorescence. But here the advantage is twofold. One is, one in confocal, what I showed, although the beam, if in a cuvette, if you are sending the beam in confocal, you are exciting actually all the plane, although you are taking fluorescence from this point, because of scattering, upper part is actually showing more light, because once it goes down, it scatters less. So here, you are photo bleaching, doing very high photo bleaching. although you are taking the emission from a tiny point. But in multiphoton, what you are doing, because the density of the photon is very high in the focal point, so you are only exciting the molecules in this point. As I told you, it is important to give two photons, successive photon, very fast in to the same molecule. So you have to have a very high density of photon. So what is possible only on the focal point. So you are exciting the molecule here, not all the other plane. So your photo bleaching is significantly less in other plane. But you still can do a lot of photo physical damage, and because of it gives high power. So, and also there's a chance of laser ablation and other thing that I'm not discussing in much details. But this is one way to overcome the photo bleaching problem. And one can do a different way. So, okay, there's one important difference between these two cases. In confocal, you need a pinhole, but here pinhole you don't need it because you are exciting only the focal plane. You are not exciting the other plane. So this is a very big difference between confocal and multiphoton case. Like you don't need a pinhole. And second one is your emission is redshifted, but here because of the how it is exciting, you are exciting with higher laser wavelength, and emission is in the fluorescent range. So one can do the same thing rather than using a single kind of single multiphoton beam. You can make array of multiphoton beam, and you can scan it, so you can reduce the time. And because it is tiny, it makes a tiny spot, which is this is a very equivalent version of spinning disk. But because you are having a very tiny spot of excitation, you don't have a crosstalk between those spots, but you can still scan it. So then I will be talking about a technique important for tissue imaging is light sheet based technique. What I also work sometime. So in confocal, you are exciting the same beam, and you are collecting the emission through the same path. But in light sheet, what you are doing, you are sending a beam from the side, and with a geometry such that you make if your sample is in the middle, you make a tiny profile here, and you collect the emission there. So if you want to take a plane image of a plane, you just illuminate only that plane from side illumination and collect the whole emission straight. So it gives you few advantages. One is imaging is very fast because it can go almost as camera speed. You can easily get 5 to 10 milliseconds per frame. So, and second thing, so this is the this is the way how it works. So you you're moving the beam, and you are seeing the whole plane. So this is one way. It's called digital scan light sheet. So you are scanning the whole plane with a very tiny needle in this case, and you can it's a one version of earlier module of light sheet. So you can image plane by plane, and you move to the next plane and next plane and image the whole plane at a time. So this is one of the movie what I showed earlier. Here you can really see an embryo from this particular movie. I have taken image from 4 views, like because the geometry of your sample mount like this, you can also rotate the sample, and you can try to get multiple angles. So in this case, I took millimeter image from 4 different views, and whole thing is done within 2 minutes. So every 2 minutes I take this kind of movie in 4 different views. So the problem is you get a huge amount of data, and data analysis and other things are also a second level of problem. Like in this particular case, in 5 to 6 hours of imaging, you can get image of the order of 4 to 5 terabyte. So that is another thing, and that's why what I mentioned earlier, computation is becoming very important. Otherwise, you can't track those cells and do the biology and understanding the biological processes using this kind of techniques. So compared to confocal, light sheet gives one advantage, that is your width, beam width, because your illuminated point spread function is like this, it's narrow. So your although the detection widespread function is much elongated, your effective point spread function is much isotropic, whereas in confocal your excitation and rejection both are having similar thing. So if you see the exact profile in confocal for this kind of low numerical aperture objective, this kind of 1 micron bead, you get very poor contrast in Z resolution, whereas you can get a very much better contrast, much better resolution in exact profile of in light sheet based microscopy technique. And second thing is the photo bleaching, because you are not doing illuminating all the plane, you are just illuminating one plane and taking image from that plane. So I'm just giving the screenshot. You are having the movie, you can check it later. I'm not going into details because I'll be going out of time otherwise. But you can have there's a comparison between DSLM and other fluorescent microscopes. You can have a look. So then I just wanted to put a small note that it doesn't stop here. You can go into tissue level much higher, like this is a tumor of BCAS, carcinoma, and here this length scale is 2 millimeters. So you can see it's a very thick tissue, and it is like millions of nuclei and cells and are there. In the earlier days, biology was more into visually seeing it in understanding. Okay, this part is cancer and this part is not. But now we want to go in details of some of this area. If you see, so you can stain them. It is by staining, HE staining. You can see the nucleus, but you can go further. You can quantitate each of the nucleus, and like that, for same sample, you can do a similar sample. You can have a quantitate each of the cell, its shape, size, and morphology, and also do a similar thing in the fluorescence, similar sample. So this is beyond millimeter. It is centimeter scale tissue. Or I'm doing another project in human brain. There you can quantitate each of the objects, and so yeah. So this is the detection where you can go in the larger domain. But of course, you can't keep the tissue intact. You have to make thin slices and then stain it and do understanding in much bigger XY plane. Okay, next part is going in the other domain, which is super resolution domain. And so how to beat diffraction limit? As I told, in this position, there is a big thing like you are limited by wavelength of light. So if you are having one molecule, you can image it like that. Of course, this is XY resolution wise I'm plotting. You can have a XY profile and you can get the emission like this big. And if there are many molecules very close by, you get a blob. You don't get where the individual molecules are located. And so how to get it? So there are techniques like spectroscopic techniques, what I won't be talking in this one. But I will be mainly talking how to get diffraction limit by imaging methods, which is called super resolution methods. And so there are two ways. One is typically called near field super resolution, which is TURF. Like what I talked about fluorescent microscope, you are illuminating all the plane. So molecules in all the planes are getting illuminated and also contributing in the emission. But if you are sending the evanescent field, I'm not going into the theory of evanescent field, but main thing is if you are if you send a light from coarse to sample with a higher to lower refractory index, beyond a certain point it doesn't penetrate there. If you change the angle beyond critical angle, so basically it get reflect it goes to total internal reflection, but still just above the cover slip there is a infrared wave, it stays, and what decays exponentially in a very close to the surface, which is of the order of 100 of nanometer distance. So you get a very high resolution because you illuminate only a tiny plane above the cover slip. So if any problem is you are addressing is just above the cover slip region, like a cell attaching to the cover slip, or some of the fibers, structural proteins, localization. So these kind of techniques are very commonly used, very powerful techniques, because it gives a very high Z resolution. It has a little poorer XY resolution, but because of your selection in Z is very high, you still have a much bigger advantages. So second, so the method what typically used in the lab is using the same objective for both excitation and emission. So what I told, if you are sending the beam like that, you can like a focus in the back focal plane, and you can illuminate all the plane, which is like a fluorescent kind of method. But if you are sending the beam from the side after some time, which is the once it's cross critical angle, it just illuminate the plane above the surface. So you can get a very J sensitivity is very high. So next is the far field super resolution technique, which is developed by these two guys and got a Nobel Prize, Herewith, Stefan Hell, and Bernard. So these are three kind of complementary techniques, and there are different versions as well. State, structural illumination, and localization microscopy. So these are very different kind of complementary technique. And again, I'm not going into details. You can go through details, but you advantage is, state, you can have a much narrow XY profile, not in Z. In structural illumination, you can reduce your Z resolution better, 3D structural illumination, and also moderately get a better XY resolution. And whereas localization microscopy technique, where you get more point by point molecules, so you don't exactly get a 3D. Okay, there are 3D palm and storm. I'm not going into these details, but you get the localization very precise way. You can go around like a 30 nanometer, that kind of length scale. In structural illumination, the concept is very simple. If you illuminate the flat hole, because in epifluorescent, your illumination plane is flat, so all the molecules in the plane are getting the same light, same amount of excitation. But if you don't do it, if you create a structure, and known structure, not arbitrary structure, like a periodic array of beam like that, sinusoidal pattern, generally in these kind of cases, you create a structure of excitation, and then you can modulate the structure. This one common way is you can rotate the structure in very fine angle, and take multiple images. So of course you will lose resolution in terms of time, because you have to get many images in the plane, and then you can reconstruct it back to get an image which is much higher resolution. The principle is very simple. Your sample is unknown, but your illumination is known, and then you take the image and do a very fine rotation, and you can achieve a much higher resolution. So that is one way. But again, I'm going close to out of time. So there are many other techniques as well in structural illumination. Like high-low is one of the method. You can rather than taking many rotation, you take two images. One is this kind of array, another is flat emission, and take the frequency component from those. You so from frequency separation, you can merge the images, or you can take only 3 images, 4 images like that with a certain phase shift in the same plane. You do the phase shift, and you can combine those images, so you can still don't lose so much time, because you limit your imaging with 2 or 3 images, but you can still gain moderate higher resolution compared to April fluorescent microscopes. So next state, the principle of state is very simple. Suppose you are having the beam of excitation like that, but you do the masking of the emission like this red donuts type of shape. Basically, all the emission is coming from all the area, but you are masking the outer part and allowing only the middle part, so you are having a much better resolution. So that is the thing. You excite the molecule, but you create a donuts type of beam overlaying exactly on the excitation beam, such that all the molecules which are outer plane are depleted. Only the emission is coming from fluorescent emission of desired wavelength is coming in the green one, which is the middle one. So from there you are enhancing your resolution to there, because as I told, the paintbrush earlier we were doing a thicker paintbrush. Now I'm going in a much thinner paintbrush, which is this green inner ring. So you can get a better resolution. Like that, if you are having many molecules like that, if you are doing excitation of this kind of big beam, you will be illuminating everything. So you won't be able to resolve this molecule. But here, what I'm doing is creating a donut beam, such that you effectively collecting the emission from this area. So your resolution enhances. Only these molecules will give the emission which is coming to your through your filter of your desired wavelength. So here also another thing, you can tune the shape of the donut to get a better resolution. You can get much narrower region. But there is a big compromise, that is the photobleaching. What I told, the depletion actually is shifting the emission from the other part in the higher wavelength. So you are not collecting that emission. So you are basically contributing in the photobleaching. So it gives a very, very high photobleaching, but still, you get a better resolution. So how donut shape beam is created is by using this kind of faceplate. So I'm not even going into those details, but these are the pros and cons, what I already explained. So next is far field localization microscope. There are multiple methods. So basic idea is, if there are many molecules, you will see this kind of block. But if there is only one molecule, you can find the center of the molecule and get the localization. So it is like that. You are having an array of molecule, but at a time you are illuminating like a switch. You are doing a switch on off, like each molecule is tied with a switch box. Like you are doing this randomly, certain molecules. So you get this molecule, find its localization, mark the point. Okay, 4th switch is on, then it is 101 switch is on, like that. So now you know which localization, which is the position of those molecules, and then you can repeat the process. So next time maybe this and this molecule will be illuminated, and then you can get the position of all the molecules ideally. So it is like there is a way, multiple ways. So one is activation or for photo switches. So in PALM, you do photo activation laser. You are activating a certain molecule in a very using activation laser in a very low power, and then use the laser, fluorescent laser beam to get the localization of this molecule. So from there we get this. In the next time we repeat it. Another few molecules will be excited. Using this laser, you can scan it. and repeat it and get it. Like that, you can reconstruct the whole thing. And same method in this is PALM. I'm not going in the details. So, but in a PALM, what you do is you are having the photo activation laser like that, and so this is the fluorescent laser and the photo activation laser, and using it simultaneously, you can get a high resolution imaging. And next is STORM, which is very similar, except there's more biochemistry involved. You need a conjugate molecule. Like if this is your molecule of interest, and you keep a molecule like a size 2, and this one is your emission of interest. So what you do at first, you inactivate all the molecules, all the Alexa 647. Then very low power laser, you stochastically excites side 2, such that it activates because it is touching each other, Alexa 647, and then using the corresponding laser to image those molecules, and then you repeat the way what I explained earlier. Exactly, it's the same method. So I just wanted to go brief, so I'm a bit rushing it up, but you can read it. It's more well explanatory.
So 1 or 2 more minutes. Sorry to interrupt.
Yeah, sure. Yeah. So you are having. So maybe you can from the movie you can get these references. Some of the papers I noted might be good for super resolution. And there are many things what we didn't explain, which is important to mention. There are level-free imaging techniques like X-ray imaging, infrared imaging, chemical imaging, electron microscopy. There are branch of spectroscopy techniques like a FRAP, HCS, FRET, and there are force microscopy like optical tweezer, AFM, and others. Yeah, but you should know these. There are techniques and what can be used in different things. So I tried to do like a different kind of problem. What kind of methods are generally used? Like a single molecular level, like DNA, protein, all these cases. We can get force spectroscopic method. There are certain fluorescent methods, and what kind of application maybe. Then there are things like on a particularly on the plane. What are the methods can be useful? Like a molecular clusters on a cell membrane and other things. So you can, from the video, you can take this, go through the details, and please feel free to email me if you have any question later. And also, whole cell level, generally this is confocal, wide field, spinning disks, and light sheet based methods. And there are methods like a tissues, the collective cell dynamics, and there are different kind of force manipulation methods. And there now you are shifting towards confocal, spinning disk, light sheet, and also some cases multiphoton. Once the moment you are going in tissue level, you are completely going out of wide field based method, and confocal, light sheet, and multiphoton is the only method. So yeah, this is the talk. If anyone is having a question, I'm having very less time, I guess.
Thank you for such wonderful talk. In this teaching talk, you have covered nicely many imaging techniques. Students and whoever, if you have any question, you can put in the chat box. Yeah, Vidisha ma'am, can you speak?
Yes, Dipanjan, it's always a great pleasure to see your slides and hear you. Oh, there's a translation happening. Okay, Dipanjan, what do you think is the best method which in not just spatial resolution, but when you don't want to compromise with temporal resolution also? Is it STED? Is it STORM? Is it 3D STED? What is your
in super resolution domain? You are talking about that domain, right?
Right.
Differential limit, yeah.
Yeah.
So in that domain, everything has pros and cons.
Yeah.
I would say if the fluorescence photo bleaching is not a big problem, then you can. But, 1 minute. Okay, sorry. So if photo bleaching is not a problem, I would say a state might give you the fastest imaging. Because at the end you kind of scanning with like a normal confocal almost, because you are having a donut beam. So that will give, but here the photo bleaching will be high.
So yeah, okay, fine, good, okay, thank you.
Yeah.
We are running a little out of time, so therefore we can have a maybe.
Okay, another thing, another just one line. It is maybe a lattice light sheet is another useful, but it is a expensive one, and probably not
cover it,
commercial.
Right?
Yeah, I have not covered it. Yeah, I didn't cover it.
Okay, thank you, Dipanjan. We'll get you again.
Okay. Okay.
Thank you.
Thanks. Bye.
Yeah. Okay, so if someone else has question, they can directly mail it to Dr. Bhattacharya.